Okay. Can you hear me?
Yes.
So let me share my screen. Does that work? Okay, let's give a couple more minutes for people to come in. Yeah. You hear me clearly? I forgot my laptop, so I set up a new system just before the meeting. It's all clear?
Yes.
Okay. And we have Sarah, which is great. Because she said she had some delay. Okay, so the goal of the meeting today is to discuss analysis of SLAP2, and I've coordinated with a few labs that did analysis of the pilot data we've collected. And so in just a few minutes, I'm just going to summarize where we are in data collection. Then we move on to discuss analysis from others. I think Alex Meyer is going to go first. Is that right, Alex?
Should I go right away?
And then Sarah, and then Nicholas, and then I think Lucas, if there's time, also said he had some things to show on statistical analysis. So in terms of data collection, and hopefully we keep some time in the end to discuss. I think it's important we keep some time in the end to discuss future experiments. So where we are in data collection, we have collected three experiments. All of these were done in the same animal. This animal was labeled with glucinifor 4. So we are imaging input to a single pyramidal cells. We record apical and proximal dendrites during oddball experiments. Boost experiments are pilot, so we are hoping that some of this analysis will inform future experiments. That's why we did this so quickly. Right now, we are doing an experiment where with the same glucinifor 4, but now adding calcium onto the cells. And so this experiment that we did last week, and we're going to probably repeat it again this week. And so I think it's important that we inform boost experiments with any analysis that you guys have done. Okay, so I'm going to hand it over to, I think, Alex, you wanted to go first before Sarah, right? So why don't you take it from there?
Okay, so the reason I wanted to go first is that I feel I've done very little and also very basic analysis. So the two things that I looked at, and you can find the code on the discussion forum where we discuss the initial analysis. The two things that I looked at was the oddballs themselves. The only issue there was, and I think that's an open ticket, that the current data does not tell you exactly what part of the experiment was the oddball experiment. So all the data is collected, and you have to, using the code or using the data, find out, was this orientation tuning, was this oddball, and so on. So that was the only obstacle there. But once the oddball block is defined, it's quite easy to find the 3 types of oddballs. Just as a reminder, this oddball paradigm is a stochastic one. So there is a repeated presentation of one and the same drifting oriented grading, and then stochastically, once in a while, there are oddballs, and there are 3 types. One is a static grading, and then 2 of the other oddballs are different orientations. And so there's 3 oddballs that basically interrupt this repeated sequence of the same stimulus. And so there's obviously a whole bunch of ROIs. And so the code that you can find there pulls out the data from individual ROIs, and you can look at each of these synapses, how they respond to the oddballs. And the code also produces a population average where all of the ROIs are averaged. I looked at mean and median, and they give you pretty much the same results. It looks, from what I can tell, and so this is maybe worth discussing, that the main effect that we find is just orientation tuning. So with the exception of the static oddball that we'll talk about in a moment. So one of the oddballs gives basically no different response to the regular stimulus. So there doesn't seem to be in the population average an effect of the oddball. So pretty much the same response for the standard response or the oddball. Now the other oddball that has a different orientation gives you a larger response, but that is probably just because that falls more into the orientation tuning. The static stimulus also gives a different response. And so that's also something we might want to talk about, because again, we are conflating different stimuli with the fact that they're also oddball stimuli. So that's, long story short. You can find the code together with the figures in the discussion forum. It's a Google Colab document. So it's a Python Jupyter notebook. The benefit there is, if you're not familiar with that you have the comments and the code, you can just click on the play buttons and it will run the code in the cloud. So you don't have to worry about installing prerequisites or anything else. Google Colab is free. So all you have to do is register, you get full access, and you can run the code. You can edit the code after you cloned the notebook, and you can also just take the code and expand on it yourself. So that's the oddball paradigm. As I said, the one thing we might want to think about there is that we're conflating orientation tuning with oddballs, and that seems to be an issue that we're already running into, or we're conflating the difference in the static versus drifting condition. So that's very precursory. I've done nothing else. I know Sarah will talk about the moment that she looked at spatial distribution, and there's other things that might be worth looking at. And I think also looking more at individual ROIs that might show an effect of oddball that doesn't seem to apparent in the population. The second analysis is orientation tuning. So most of you are probably familiar with that. Neurons in V1. If you present them gradings or line orientations, they respond to varying response rates, various magnitude. And so there were 2 blocks in the experiment where gradings of different orientation were shown in random sequence. And so the code that I wrote looks at both of these periods and then computes the curve of responses as a function of orientation, and plots those in a polar plot. So you can basically see as a function of degrees, you can see where the tuning is at. You should keep in mind that that's a 360-degree plot, but we're looking at line orientation, so it's only 180 degrees of orientation tuning. Nonetheless, the tuning sharpness, so that's basically how much you can change an orientation before the response significantly drops, seemed quite sharp for most of these synapses, most of these ROIs, and they also showed a pretty large overlap. Now, I haven't published this code yet. I will do so as soon as I can, because the plotting function is still a little buggy. And so from what I learned so far, that's not a bug in the code. It's part of the matplotlib library. So I try to fix it. It particularly affects the population average tuning. But if I can't get this fixed, then I just release it as it is. And so I hope that other people will be inspired to look at that, and then feel free to just use the code. There's much more to do, right? So with the orientation tuning, to look at the spatial distribution, synchrony, all of these things. So that's it.
Okay, thanks, Alex. So should we? So I know Sarah is Sarah here. She said she had a lot.
She's on the bus still.
Yeah. Okay.
John,
was
can I
one
ask you a quick question? I have a quick question. So I was checking the Python notebook on the website. So there I see 58 ROIs. Are these 58 on the same neuron but different parts of the dendrite?
Yeah, we're imaging boutons. Is that what you meant?
Yes. So there are 58 boutons on one dendritic branch of one neuron?
They're distributed across the cell, and so we are imaging the apical and the proximal dendrites. Some of these boutons are in the same dendritic branch, some are on others. So yes.
And then how do we know they all belong to one neuron?
So that is careful tracing during the experiment by Maide and Andrew. And we also acquire a stack, but I feel fairly confident that those dendrites belong to the same cell.
And they are apical dendrites in layer 1? So they're
Yes.
apical dendrites of but layer 5 neurons?
Those are layer 2 free cells at the moment.
Okay. And this is V1.
This is V1.
Thank you.
Yeah, so one thing I wanted to say is because, again, because I'm trying to advocate for using Jupyter notebooks, that, for example, the Jupyter notebooks that I published, they keep all of the links at the top because we have the information a little bit distributed. So your question was, for example, what are these neurons, where they imaged? Another question is, what was the exact stimuli that I used? So there's one link that goes to the stimuli. There's one that goes to the experimental details. And there's another link that explains the overall design.
So is this now here? Because I cannot, I'm on the notebook, but I don't see these links. Or are you saying that the plan is to add those links?
It's at the top of the Google Colab. So there's right underneath the title. So basically, there should be links to the NWB data, there should be links to the experimental notes, and there should be links to the stimulus itself. So the idea is to keep this together with the actual data analysis, so this can always be reconstructed. Because obviously, as we're getting more and more data coming in, I think it will be important to keep track of all of that.
Okay, I will have to write you separately because I'm not able to find it. I'm on the Neural Dynamics GitHub, and I don't see this that you're saying.
So it's not on GitHub. So if you go to the discussion section in the GitHub, and we talk about preliminary analysis, if you scroll down, then one of the posts is a link to the Google Colab. And so that will take you onto the Google website, and that's where you see the notebook.
Sure. Okay, where is the discussion section on GitHub?
You click discussion, yeah.
There's discussion, I see experimental summary, stimuli, hardware, templates, analysis, experiments, but I don't see discussion. I see
I think the one at the top, I don't have it in front of me right now, but I think the one off the top says something about SLAP2 analysis. And so that you want to look into that discussion.
Okay, I see overview, experimental plan, analysis plan. I don't see discussion.
No, you're in the wrong. Let me share my screen.
Thank you.
Okay, so you're on this website, that's right?
That's exactly where I am, yeah.
If you click on edit on the top right.
Okay.
I need to log in, but
yeah, it brought me to GitHub, to actual GitHub. And then I go to discussions there. I see.
website.
So I have to go to the actual again. Yeah.
Yeah, sorry, I don't want to edit. Oh, and because I'm on a new computer, I can't log in. I'm sorry.
So maybe I share because I do have it actually on
You
my
have it?
thing.
Okay, thank you.
Yeah, so let me share that. Okay, so I'm here. Well, I did what you said. So
Yeah, you got it.
so
You're
exactly.
on the
So I
farm.
go right. So I go here. No, I came out of it. And then I go to discussions,
Yeah.
and then where is so when what Alex was saying, that's in here.
As opposed analysis. Yes.
Just scroll down.
Okay.
So there's the blue, right there, the blue one. Oh, scroll up a little bit, right above the figures.
Yep.
Right there,
Okay.
that link. If you click that, so now you end up in Google Colab, and there you see there's all the information right there, right there. You see all of these links.
Amazing. Yes, I see
So
all of this. Right.
Right, what you're looking at. So there's the
Okay.
notes.
So to get there, we have it's a little bit convoluted, all of that, but I guess that's what everyone has decided. So is there a way to add this to the GitHub to that? How should I refer to this page? I mean this, not this. Sorry,
The repository
this.
discussion forum, Farzaneh, is I think
Yeah.
the best place to have conversations on these things.
So you prefer to have those there?
Yeah, because
the
website is a fixed representation of the project at a particular date. And if you want to discuss individual page, I think we should use the forum.
Ron, can you add a link to the forum to the meeting, the meeting invite?
Yeah. I will add those things.
I would say minimum it would be helpful to have a link to the repository on here.
Yeah, we can put it under analysis, right? So there's analysis on the left-hand side. Yeah,
exactly.
I can do
here.
that.
Well, actually, I think
any page actually has a link. So if you go to the website,
then what is this, for example? So when I click on this, how is this then related to the other things?
So yeah, so we haven't talked about this one. This was the very initial data analysis, was actually done by Jerome. So what you see on there are Jupyter notebooks. One was done by Jerome and one was done by forget your name.
Carter.
Yes, by Carter. So that is code that we then built on. So what this code does, it just generates very basic information about the experiment, but it's a good starting point. Maybe we should document all of this a little bit better. Yeah.
Yeah.
Okay.
And then if you go back one to the website, there's a start a discussion buttons in front of you.
Yeah.
You click on that, it brings you to the forum. That's a new page on the forum. So any page can be discussed this way.
Okay, and then I have one more practical question here. So I have students who are interested in analyzing this data. Where should they start?
On the forum thread that.
So they have to go through.
That's right, because I think we have to coordinate ourselves, and you have to know what's been done.
Okay, and then where is the data being uploaded?
That's this, but I think Farzaneh, you should spend some time reading the website. I think there are a lot of information already.
And when you say website, you mean this? You mean
no,
this?
No, the static website, we have pages that describe how to access the data already. There are meeting notes for individual experiments. I would.
This is where I need to spend time here.
Well, if you have not, yes, but I would.
No, this I guess I know. I guess
experiment summary.
Okay.
There are yeah. So this are the experiments that we
okay,
did.
Thank you. So it's in the experiment summary page.
There each individual, yeah, there are access to the data. I think it's been documented there.
Yeah, thanks. I mean, I have gone to this many times, but some of these categories are not as intuitive. So I mean, so what you said, and it's super helpful to me, is that this is where I need to go, and then that's it.
Okay. I'm happy to make it more intuitive.
No, I guess it's fine. I guess with some of this, it's just the
I want to make sure that we discuss the analysis, Farzaneh, if that's okay.
Sure. Yeah, please.
So Sarah sent a PowerPoint prior to the meeting, and so I was hoping to share it and go through it. I don't know if I think Sarah's probably offline, so we can maybe unless you can join. Can you join, Sarah? Oh, we can't hear you. Ah. Alex, do you think you could help
Sure.
comment on the slides?
Yeah. So the first slide we're looking at here is at the top, what you see is the entire experiment that so far we chose to collectively analyze. So on the top, what you see is stimulus orientation of the gradings as a function of time. And it's colored because what I said before, that hopefully in the future we have in the data very precise points of what is done what. But what you see there is in blue is the orientation tuning. And it's colored because that's how we detect it. We basically just write in the code, show us in the experiment where the orientation changed a lot, and there's a whole bunch of orientations. So you see that the experiment starts out with orientation tuning, where it's just a whole bunch of different orientation gradings that are shown. And then what you see in red, the longest block of the experiment, so those are the oddballs. And you can tell because the line is almost flat. So it's almost always the zero orientation. And then once in a while, you get these spikes because these are oddballs of different orientation. Right after that red block of the oddball experiment, there's another block of orientation tuning. And then you see very last, there's another blue block where you see that there's an oscillation between certain orientations. And so this is actually the receptive field mapping that was done at the very end. And so this is basically, there's three parts to the experiment. One is repeated. And so this is how we detect it. Now, below this depiction, you do see the different oddballs within the oddball block. So you can imagine we're zooming in. And now what we're color coding is the three types of oddballs. So there's two different orientations. I think it's 45 and 90 degrees rather than 0. And then there's also the static grading. And as you can see, it's fully randomized. So most of the time, the subject sees 0 orientation, and then once in a while, there's these oddballs, and they seem to be randomized. Does this make sense?
Thank you, Alex. Let's go through Sarah's slides, and I think you're best to comment, probably, Alex.
Can you go to the next one?
Actually, I have a question, actually.
Yes.
Can you hear me?
Yes.
So that final block at the very end of the trial, what exactly is that? It looks like the orientation. I'm just a bit confused about what that final part of the experiment is. Where we see that blue block.
The receptive field, that's receptive field plotting. So what is done there is there's a very quick succession of showing static gradings in different parts of the monitor to find out where each of these ROIs responds to in terms of location on the monitor.
So there are three orientations for the receptive field block. So we're displaying tiny drifting gradings that the orientation changes to maximize the chance of seeing a response.
I see.
And I think it has to be different. I think it has to be different orientations, Jerome, right? Because you don't want to accidentally show one that the ROIs don't respond to. So you have to sample different parts of the orientation tuning.
Yes. Okay. We're going to add some more explanation of this. Clearly, that's missing.
Can you go to the next slide? I think we're still looking at slide number 1. Yeah. Who's sharing that screen?
I think, Jerome, you're not. It's not updating for some reason.
Really?
It might. I think you're sharing the wrong screen.
Oh, maybe. Okay. This is funny.
You could probably just drag one to the other or something.
Does that work now?
Yeah, that works.
Okay, so this is from the original code from Jerome and Carter. And so what this shows are the two slices within Cortex, or the two horizontal planes. They are called DMD1 and DM2 in the data. And each of these images is indicating the location of one of the synaptic boutons. And in there, it's actually a tiny plot that I believe shows the receptive field.
No, I think it's just the location of the ROI.
Okay.
I think, and then the sum of the mask. This is just the strength of the mask. It's just to position the ROI in. Yeah, that's what Sarah had it is. So this is this is addressing some of Fazane question.
And so which one is higher up? Do you remember, Jerome?
Which one is what?
Which one is apical? Which one is higher up in cortex?
I think it's DMD2. Yeah, I think it's DMD2. This is DMD1 is the somatic.
That was what the website says, I think.
Yeah.
Okay, so then we can go to the next slide. So just to repeat all of this, the code is available. So this is something that Sarah extended to the right-hand side. You see the oddball responses. So that's what I described before. So these are the what we're glutamate imaging, right? So it's the glutamate signal. And so you see at 0, the stimuli come on. The good indication here that all of these different conditions, they overlap before the stimulus comes on, which is obviously what we would like to see in a baseline. And then with some delay, this is on top the DMD1 average. So those are the ones that I believe are somatic, right? So and what you see in gray is the black line rather is the static oddball. In green, you see the oddball of 90-degree orientation, and then the remaining two conditions, the faint gray line and the red line, those are the red line, the 45-degree oddball, and the faint line is the standard stimulus that is repeated most of the time. And you see how the red line is right on top of that gray line. So that is what I meant before, that doesn't seem to be much of a deviation for the oddball. And that is also true for the other plane. So that's shown right below the DMD2, the one that seems to be more apical. And you see the average response right there. Now, the Z-scores are rather low. I think Sarah is ready, so maybe she can take over.
Why do we not see much response to the 45-degree? That's very cool.
I think it's the orientation tuning. So from the tuning curves that I looked at, it's basically a stimulus that these boutons seem to be very little interested in. But yeah, it's
they're spines, not boutons. Just for sorry to interrupt. I would be that's actually kind of surprising. I mean, maybe Mada can chime in. I don't know. We don't see a lot of neurons that like. Usually, we see very heterogeneous tuning. So if there aren't specific orientations that inputs tend not to respond to in our experience. Is that true here?
So there's a population average, and I did see some differences if I took mean or median. So I didn't spend a lot of time looking at the heterogeneity. So this is just simple averaging everything together.
Okay, and so you're saying the inputs do have a preference, and it's away from 45 degrees. That's worth Mada. Maybe it's worth looking at that. You as well. Mada has a lot more context from other recordings. Thank you.
Yeah, sure. I think I missed this part. Why do we see like 29 ROIs and 13 ROIs for the DMDs? Like, I'm sure you explained this, but I think I missed that part.
Yeah. So one thing I should emphasize. So I think we have more ROIs, but so one thing I should emphasize is the code is preliminary. So for example, if there are some issues with the data, and there's NANs in the data, for example, then I just kick it out and move on just to get things done very quickly. So it's really worth going over all the code that we have and don't see it as gospel, but go line by line and see that, wow, actually there's some fudging going on right here, right? So you could, for example, interpolate NANs and then probably rescue some of the data.
Yeah, there are a lot of NANs in the data. We are very conservative about discarding data where there's a risk that there was motion, for example, or things like that. And so we inject a lot of NANs actually into the data where we got recordings, but we just want to be very conservative there. I think it's very important that analysis methods can deal with, basically brief periods of NANs in the data stream. Even very high quality recordings will have NANs for this reason.
Yes, but those NANs will be similar for all the ROIs within a DMD. So I guess I'm a little bit confused as to why here we are only looking at 29 ROIs, for example.
Can you guys hear me?
Yes, Sarah. I please.
Okay, so the reason is very simple because I basically thresholded the ROIs according to some kind of very low threshold z-score, which is why I wrote in brackets z-score above 0.5. And so I tried to eliminate some of the ROIs that had no evoked response to visual stimuli, basically. And I think Lucas will talk more about this. It's basically trying to sub-select ROIs based on some response criteria, right? Are they responsive or do they have certain properties? And basically just sub-selecting based on a positive modulation above 0.5 eliminated a bunch of ROIs. And if you look on the left, you have a cumulative distribution of all the ROIs that were fed into this data analysis for this particular recording. And so you see there's a small set of RIs who have a negative modulation, but a good chunk of them just has a very small modulation in terms of the z-score. And so we mainly got rid of these. And there's one of the 3 recordings which has a higher z-score than this one, but 2 out of the 3 are in this realm of a maximum z-score of around 1, I would say. Which is why I said we have to maybe define criteria on what are the spines we basically want to focus the analysis on. But I just put in a random threshold of 0.5 now, which is not very stringent for now.
I see. Thank you. Yeah.
Why? Okay, so just speaking up on Mada's behalf, especially, we have many recordings where there are hundreds of synapses that have extremely robust responses to different orientations. I'm looking at, I can show a movie of these orientation responses of synapses. So I'm not sure what it is about this recording that would have so few synapses at a given z-score. If you compute things like and I mean, I guess that's what you're doing. If you compute like the average orientation response, can I just show, Jerome, can I just share something for a second?
Yeah.
Just for context. So, right, this is scrolling through, just going through synapses in one of these experiments. The individual synapses have very sort of, varied, not everyone responds, but they have these very heterogeneous responses to these motion stimuli, and there's hundreds of them. So perhaps this particular recording is poor, in which case we probably shouldn't devote that much effort to it, or maybe there's something that we're doing wrong.
So I looked at all 3 files, and the highest z-score I got out of the 3 was one that had a distribution that ended at a z-score of 2. So maybe it's something in the way we calculate it, which is different.
Yeah, so can I ask you quick? So one thing came up was that with the delta f over f or versus f. So I worry because my code, for example, does not do any detrending, counteract photo bleaching. So then maybe that could bring down the z-score if you average over many trials.
Yeah, so I think what's in our current files, I don't know what you're just showing, Casper, but what's in our current files that we have on Dandy is just df. So that might be a factor.
Yes, we should just sanity check that people are working with data that is of the quality that we expect. That's all I'm saying. That might require Mada, you getting involved and just running your processing on some of this data as a sanity check of the data quality.
Yes.
It's possible that something's happening in the packaging. I don't know. There's sort of these are super early days, and it's great for everybody to be collaborating. But we should just make sure that we're not that like all the early stuff is happening correctly.
Okay, let's just compare the metrics with other data assets.
Yeah, I can just go through this data in my code and then compare the quality of this recording to my own recordings and make sure that everything's all right here.
But I remember being in the room thinking that it was an excellent recording, right?
Yes. From all I know, these recordings, if not better, are the same quality as my own recordings. But I'll still check to make sure.
And maybe we can set a standard. So for example, should we do df over f, and should we do detrending, and what kind of detrending should we do?
That's all been done. Oh, sorry, Mada, you go ahead.
No, I just wanted to say that I mainly work with DF only at the very end after doing all the averaging. Maybe I do some normalization and I do not discard any ROIs because I think in the pre-processing pipeline, there are still some threshold to discard things that don't make sense. So I think my first, like maybe my preference is to include all the ROIs and then if I don't know if it doesn't look good at all, then think about thresholding based on some criteria. Because I want to say like 29 and 13 is just a very small number compared to what we image. And again, from like my expectations, I get like hundreds of these ROIs, and I want to say maybe 80-90% of them are actually good.
And what about the trending?
What do you mean by that?
Photo bleaching. So if you look at the signal, you can tell that it, yeah, so do you do something about that?
Not at this moment. I think in the pre-processing, Casper, it's just the baseline. We count for the baseline. We don't correct for photo bleaching, I think.
Yeah, it's worth, let's spend like 3 or 4 minutes talking about delta F over F and whatnot. So we record a raw fluorescence signal and we fit the trend, the baseline F0, and we remove the trend F0 and we save that F0 as a separate field in our data files. And then we also compute delta F over F, which is the delta F divided by the F0, right? So all of those things are available in the data files that we generate. Carter and Jerome sort of made a quick and dirty packaging of our MATLAB data files into the data that you're working with, which is missing actually quite a bit of the stuff that we package in the MATLAB structures. And so we're working on fixing that. And that's, many apologies there. The correct thing to do is not to work typically. So as Mada alluded to, it's not typically to work directly with delta F over F because you lose the information in the raw photon space about like what you have more confidence in, etc. The correct thing is you need both the delta F and the F0 typically to do most things. And we do a lot of stuff basically, as Mada said, using delta F, and we aggregate things in the raw photon space. And then we also aggregate the corresponding F0s. And then if we want a fractional change, so you might compute some signals over the course of the recording by aggregating delta Fs across synapses, but then you'll also aggregate the F0s across synapses, and then you'll do the normalization at the end. And that's typically the right thing to do, sort of compute delta F over F only once in aggregate at the sort of end of your computation. Depends a little bit on the details of your computation, right? But remember that this is like glutamate signaling is a non-equilibrium process. You're not measuring a concentration of a thing. The measurements have variable noise depending on brightness, things like that. So detrending should only happen usually as the last step, or sorry, the delta F over F, the normalization rather, should only happen typically as one single step at the very end of an analysis. Happy to discuss that. It sort of depends on the details.
And is the data that we have that is not normalized?
I think I think Jerome and Carter packaged only DF, and they packaged it as delta F over F, which is very confusing. Yeah. And so we're fixing that, but it hasn't happened yet. Right?
Yeah, we'll fix it. Yeah. Clearly that's important. I apologize for that.
And you're suggesting that we normalize by the group average rather than by the average of individual ROIs, is that right?
I'm suggesting that you only normalize once, that you don't start by normalizing and then do any averaging, because that will average in noisy things weighted incorrectly with non-noisy things, for example, right? And there's other errors associated with normalizing first. If you're going to normalize, which is useful, that should be the last step of an analysis. That's the sort of that's the take-home message that I'm trying to say. But you should just think about how noise and confidence, when you average things, you want to average them typically in the space of photons before normalization, and then also average your baselines and then normalize at the end. So you would compute, for example, for like the plot that's shown here, you would compute the average oddball response, right, in photons, in units of photons, raw photons, unnormalized. Maybe that's already what's being done here. And you would separately compute the baseline for those same moments in time across all. You would basically do exactly the same computation on the F0 signals, on the baselines. And then you would divide the first thing that I said by the second thing in order to get a normalized response.
But then how do you get an error estimate? Across stimulus presentations.
So you can do so it depends. So you can aggregate your thing for every stimulus presentation, right? And then you can normalize at that level the aggregate over synapses for each stimulus presentation, and then you could get an error estimate on the normalized signal by computing the normalized signal for every presentation, right? And then you'd be doing your normalization at a different stage there. Alternatively, you can do propagation of errors. In some cases, there's code for like automatic propagation of errors, where the initial signals all have errors, which are actually in the data structure as well. And you might be able to just do automatic propagation of error.
Okay, because the rationale of the way I did it is I wanted to identify, first of all, get a sense of what's the fraction of RIs, which I would call visually evoked. And part of this is I want actually a sense of the trial-to-trial variability of these responses. I don't want to have just an average and then say, on average, this is different than baseline, right? And so that's why I basically did this kind of sub-selection.
Yeah, I think that's a wise analysis. With regards to z-scores and whatnot, I would just keep in mind that individual trials look very different than these averages because there are very brief, large events typically on individual trials that are highly stochastic. And so even if you had zero noise in the measurements, if the measurements were perfect, they would have low z-scores because of the biological variability of at any given moment, at any given sort of 10 millisecond window. You may or may not have a release event. And so there's a lot of biological variability. And so you just have to be careful about how you calculate z-scores and over which timescales if you're going to use z-score as a criterion. And it's sort of like it looks, when you say like almost all the z-scores are below 0.5, it's just kind of funny for us to see that when we look at the traces and they look, the SNR of the transients might be like 20 to 1 or something like that, right?
Yeah, makes sense.
Yeah.
So percent change might be the way to go then, right?
Like a peak-based SNR or something, I don't know. Yeah.
Simple percent change from baseline doesn't run into that problem that z-score has, that you're factoring in the variability of the baseline. So then these transient events shouldn't matter as much if you take a large enough window.
To me, you're saying two different things there. You're saying like if you take a large window, that means like filtering in time, like smoothing in time, or down.
No, for percent change, right? So you basically set the baseline to 0, and then you say what's the percentage of signal change. And so in contrast to z-score, it doesn't suffer from what you said, that you're factoring in that even the baseline is already very noisy. Because if you look at the baseline right here, if you just look at the average, it's quite flat. So if we take that and then we look at percent change as the response, then we should get something. Yeah.
Yeah. I agree with that as well.
There are different ways to do the z-score. Yeah. I feel like we should have this in-depth conversation in writing probably, because I think the details matter. So maybe, Sarah, are there a few slides you want to highlight in your analysis?
So I think, I mainly wrote a pipeline independently of thresholding and so on. So I think it shouldn't matter in terms of how we do the details of the percentage change and so on at this stage. I would like maybe to highlight something like the slides that are like something like 6, 7, something like this as an idea. Maybe take 7, something where we can see the numbers a bit better. Yeah, that's just nicer. So what I was trying to understand is there a relationship between the function response in terms of how similar are different ROIs responding, and can we kind of identify which ones share some similarity, and then see how do these actually map onto the spatial locations of these synapses. And so what's plotted here on top is basically a hierarchical representation of the activity, because I basically just clustered the traces hierarchically based on the similarity of the correlation of the different pairs of the ROIs. And I draw here a dashed line, which is like some arbitrary threshold again, right? We can debate where to set it to basically illustrate that you can use this to identify different clusters in terms of functionally similarly responding ROIs. And then we can use these to basically find back their anatomical location and basically visualize that at the bottom. So we're not plotting the response amplitude here. We're literally just plotting where were the neuro or where the RIs that were associated with being in an orange cluster, and they have some shared activity with some correlation value that we have now a parametric access to. And so you can see, right, some of these things are quite distributed. And I did that in a stimulus-specific way. So what's plotted here is for the standard stimulus condition, for instance. And then we can go to another slide, maybe 8. And we probably have the same recording. But this is now, for instance, for a 90-degree oddball. And so this was just trying to understand, is there some kind of basic logic at first sight that could explain whether there's some spatial enrichment for synapses with shared functional properties, and does it have some kind of tuning specificity to it? And this is just a nutshell approach to kind of build a better pipeline to really look at this more systematically. But in terms of a visual representation, I think it's quite nice. And as I said, it can be thresholded arbitrarily, right? You can say you want to have a similarity above X, or you can have some very refined criteria to basically identify ROIs with dissimilar activity patterns, for instance. And so that's the main thing I've been trying.
Okay. So you will say like you're sort of you're still in the stage of setting up the system to analyze the data. Is that right?
I mean, you can analyze it, right? But ideally, I would like to select the ROIs that I feed into this. I would like to define some threshold criteria of what do we want to call similar or distinctly active. And then basically look at this in a more quantitative way, in a sense that I can't make any statements based on one of these recordings, basically, besides that most of the synapses don't have an obvious localized clustering. Sometimes there is, right? But in terms of a pattern, I can't make any statement.
Okay. I wonder if it might make sense that Nicholas present now in the last few minutes, because I think he has some really related analysis. Could you share your screen, Nicholas?
Yeah, I can share my screen. Let's see.
I'm already getting a few take homes as you set up, so we need to compare with MADE. We really need to repackage the data, and then start and try to agree on how we decide which ROI are responding strongly. I think like we'll have to align ourselves on this.
Yes,
okay, I can see your screen.
Agree with that. So since you had mentioned in the discussion thread about the photo bleaching artifact, I decided to take a look at that. Here, I'm just visualizing the traces of a few ROIs from one of the data sets you had provided. And I followed your recommendation and applied a rolling mean to remove the photo bleaching artifact, at least as best as can be done. And here we see a visualization of that, where we see on the left subplot, we see the original raw signal in gray, and we see the detrended signal is what I call it, but it's the signal that has been filtered through this rolling mean. Of course, there are NaN values, so when you calculate a mean in Python, that can be problematic if there are NaN values. So I calculate the median in those cases where there are NaN values in the window of the rolling mean. Was there a question?
Yeah, so, okay. So the indicator only goes positive, right? And the data that you got, the baseline has already been removed. using like very well vetted, relatively well vetted, sophisticated methods. So further removing baseline is only going to make the periods of quiescence in between moments of activity, like what you see in the blue trace on the right, go negative, which is like not reflective of what's actually going on, right?
Yeah.
The indicator sits at 0 and then goes very positive when there's synaptic transmission, right? So you can see like on the upper right corner, this ROI8, what's happening is a stimulus is turning on and the synapse is getting really active. And then there's sort of an off period in between stimuli. And there's like those first 4 periods. Those are 4 like very highly active bursts of the thing. And it should be going down to roughly 0 in between. So the original gray trace, the raw there, is a much better baseline subtraction than the blue.
I see. Yeah, that makes sense.
I think maybe the other way to do this is not to, maybe correct me, Casper, but is not to correct the baseline, but to correct the amplitude.
Yeah, exactly. So there's a need to, so if you look at your long-term recordings of the hour of data or whatever, for 50 minutes of data from the previous slide, you see that the height of the peaks is going down, right? And that is something that you might want to correct and that's simply because the whole thing is getting dimmer, but the baseline has already been subtracted. And so that would just mean dividing by this F0 value that we will package for you, that it's just an error that it wasn't packaged for you already. And that will bring back the amplitude at the ends of those recordings. Though possibly it will do that in a biased way. That's the worry. Like, maybe it won't stay exactly flat. Maybe it'll get bigger. Maybe it'll get smaller. Maybe the real biology is causing it to get bigger or smaller. It's hard to know exactly, how much to divide by, and that's one of the problems. But we need to doing this additional baseline subtraction, I can say already, is probably a bad idea.
Can you clarify exactly what the F0 value is? What exactly is that?
The F0 is an estimate of how much unbleached indicator is there at any moment, or rather, how much brightness would we expect to get if there was no bound indicator? Like if there was 0 glutamate, F0 is how bright every pixel should be, or every, in this case, synapse should be, if there was 0 bound glutamate. What we want to do is, by subtracting F0, have this delta F, this change in fluorescence, be proportional to glutamate binding events. The issue is that you do need to divide by that baseline, right, as well. And the data that we've given you hasn't divided by that baseline, because dividing by that baseline then gets rid of crucial information about noise, right? So keeping delta F in photons allows you to, for example, aggregate across synapses correctly, and then you divide by the baseline only after you aggregate across synapses.
I see. That makes sense. I understand that.
We'll give you the baselines,
okay.
And then as the last step of your various analyses, you can divide by those baselines.
Okay, sounds good. So the plots I'm going to be showing now make use of the raw signal rather than the filter signal I showed in the last slide. So in this case, what I did was I took the peristimulus windows for the 45 degree, 90 degree, and standard stimuli from the pre-block, that is the orientation tuning block prior to the odd block, for the odd block, and for the orientation tuning block after the odd block. And my goal was to see how the average responsiveness of each ROI to these different stimulus presentations varied because of the presentation of the odd block. So in these plots, what you're seeing are the subtraction of the average responsiveness of each ROI to the stimulus presentations between these different blocks. In the first row, we have between the odd block and the orientation tuning block prior to the odd block, which in this case I call pre. And the second row, we have the subtraction of the average responsiveness of these different ROIs to these different stimulus presentations. In the post block, minus their pre-block, and so on. So what these colors are telling you then is how much more responsive these ROIs are relative to this pre-block. So when we calculate the average responsiveness of these different ROIs in the odd block and subtract it from the pre-block, we're seeing how much more active these ROIs are with respect to this stimulus presentation. Yeah.
So let me rephrase. So the post is measured during the orientation tuning block. Is that right?
That's right. Yeah.
I think any decrease here is hard to interpret before doing the bleaching correction, because I think it could be due to that. And the increase is interesting because I think it's unlikely that bleaching could cause an increase. And so if we see an increase in response tuning, it might be true. And so I feel like we should be cautious, but and repeat this analysis after we have done the normalization.
That makes sense to me. Yeah, that makes sense.
Yes. I just want to say that we will repeat this analysis absolutely once the data has been corrected for all these effects.
Yeah.
I think what Nicholas wanted to show is that he's already built all the pipeline to overlay the changes relative to all these different blocks on top of the spatial distribution of the actual DMDs. So I think we've, the pipeline is there. So
yes.
And there are more figures in the discussion thread beyond this as well.
And I guess some of the clustering analysis that Sarah has started can be put on top, right? So you create the visualization and then we cluster it after that to sort of highlight which one of these are reliable.
Yes.
Yeah, and Nicholas had done some similarity calculations as well.
Oh, yeah.
We wanted to pick up the community's view on how this could be approached. So I'll let him summarize what he did, and then we can discuss.
So in this case, I wanted to compare the pre-block to the post-block, which are respectively the orientation tuning block prior to the odd block and the orientation tuning block after the odd block. And to describe briefly how I calculated the metric you see visualized in these plots, I measured the responsiveness of each ROI to all the different degree orientations of the stimuli presented to the mouse. So by calculating those average responses to these different orientations, I got a vector that you could call an orientation tuning vector, where each element of that orientation tuning vector can tells you the average responsiveness of that ROI to these different orientations. I then took in this case, the 10 nearest neighbors of each ROI when evaluated in terms of its spatial distance from all the other ROIs. And I calculated the cosine similarity metric between, say, the ROI of interest and its 10 nearest ROIs with respect to their orientation tuning vectors. I then averaged those cosine similarity scores to get an average of how similar, say, the tuning vector of one ROI is to its neighbors. So then I did that for both the pre-block and the post-block separately, and I subtracted these average similarity metrics to get you this plot here, where a blue color would mean that each ROI is becoming, the orientation tuning vector of each ROI is becoming less similar to its neighbors. And red would mean that it's becoming more similar to its neighbors. So here we can, so here we can see a kind of clustering going on, at least, but that's a hypothesis, of course. It has to be tested.
Okay. So when you said less similar, just want to clarify one important thing. White color here means that they're probably orthogonal, right? But blue means they're anti-correlated. So if you have an orientation tuning to 0, then the blue would mean it's 180-degree tuning, right? So it's anti-correlated, not orthogonal.
Yeah, that would be true of the upper two subplots. The bottom plot, which shows the subtraction, would tell you that they're becoming more anti-correlated versus more correlated versus, yeah, that's what the colors in the bottom plot would be telling you. Yeah.
So we're at the hour. I just want to make sure that we have a plan moving forward before everybody has to move on to the next meeting. So it seems like we have still more content to cover because I think other people have worked on this, and so I propose that we continue this conversation next week. And the second is, I think it would be great if we could all share those slides on the forum before the meeting so that everybody can see them. And perhaps comment. So I would suggest that all of those that have done analysis share them on the forum and then comment each other so that when we go to the meeting, we can focus on discussing the interactions instead of the content exactly. I think that would be more efficient. We are making mistakes as we are packaging this code. That's what's expected because we're trying to go fast. And so I think we'll correct the issue with the missing data this week so that you can address some of the pitfalls that Casper pointed out importantly. Am I missing something important? I feel like we probably want, I would say we probably want to run the control of bleaching and so on and so forth before we make any scientific conclusions. So I would wait a little bit before we make decision. Does that make sense?
Yeah, makes sense.
Yeah.
So.
Just one question, Jerome. For the discussion of the different analyses, right now it's all in this one discussion thread.
Yes.
Which gets kind of difficult to see. Would it make sense to open separate ones for the different analysis branches, so to speak?
I would say maybe it makes sense that each one of you open a thread with your you just start a thread with your PowerPoint and look at the code whenever you can link it, and then we have a discussion on that content. I think I'll probably be more structured.
A suggestion is to, Jerome, you put the list of questions that we want to tackle first. And I suggest each question has its own thread so that we don't mix the results. Let's say, for example, someone else's wants to capitalize on Nicholas' results or, Alex's results. Then if they all under the same question umbrella, then it logically makes sense to put them on the same thread. You know what I mean? Otherwise, we can have a huge thread of all, comments and figures on all the questions, and it's going to continue to grow and make very, very hard for people to trace what's going on and who can contribute what. We don't want to be redundant. We want to complement one another.
Yes, I think that makes sense. I still feel like the thread will grow, but let's start there. Creating a thread per analysis question.
That's my suggestion, but welcome other suggestions if it's easier. The idea is to make it easier for people to find what they're looking for and not to repeat others' work.
I think that makes sense.
Yeah, maybe it works to link the other thread. So instead of posting the plots, one can make the statement to this general discussion and then link to a result in the other thread, so that it's just not it's like this overload of results.
Yeah, let's do that. Okay, so next week, let's keep the meeting to continue this discussion. I will also present some progress on stimulus development here related to Bonsai, so that everybody is in the loop of what's going on. But maybe we could do like 2/3 of the meeting on analysis and 1/3 on stimulus design. Does that make sense?
Yep.
And we'll try to repackage it as quickly as we can with the newer missing assets.
Sounds good.
Any question we should address before we report? Okay. Thank you.
Thanks.
Have a
Thanks
good day.
all. Have a good
Bye.